to introduce you our study on the effect on genes involved in neuroplasticity and environmental stress on the short-term response to antidepressant treatment. It is well known the important role of the genetic background in the pathophysiology of the anxiety and depressive disorders as on the antidepressant response. The most consistent finding is on as you can see, the polymorphism of the promoter gene of the serotonin transporter gene. So, in this study, dating back in 1998, shown how the homozygous and the heterozygous of the serotonin transporter genes had a better response to the antidepressant treatment. During the last few years, a lot of studies focused on the association between the antidepressants response and the neuroplasticity impairment. In particular, the brain-derived neurotropic factor has emerged as a crucial mediator for the neuroplasticity. So, in BDNF knockout models, they displayed an inability to respond to the antidepressant treatment, as serum BDNF levels are reduced in depressed subject relative to controls, but this deficit can be normalized by the antidepressant and the electroconvulsive treatment. But as the chronic stress can disrupt the neuroplasticity, the exposure to stressful life events may also interfere with the pharmacological treatment too. As you can see in this study, subjects with a higher perceived stress show a worse response compared to the subjects with a lower perceived stress. But the gene and environment interaction on the response to antidepressants is important too, because as you can see in this study, different carriers of the serotonin transporter gene had a different outcome in the response, and it's modified both by the gene background and the stress exposure. But when does the stress have to occur, and what will it cause? Because according to the period when the stress might take place, different structures can be affected with perhaps different consequences. So, in this study, you can see how an history of childhood stressful life events the patient have predicted and a significantly lower probability of remission at 12 weeks of antidepressant treatment. So also the stress occurring at the onset of the disease are important because, for example, in the studies you can see the response of the short genotype of the serotonin transporter responded poorly to the treatment with the fluoxetine, but only when they were also experienced a prior stressor before. Also, in the same way are the current episode stressful life events. These studies show the different response of the short genotype of the serotonin transporter gene with people reported stressful life events at the current episode, which had a worse response compared to the one without stressful life events. So the aim of our study is to analyze the impact of genes involved in neurotropic process on the antidepressant response and the impact of the stress at different time points, and mainly the potential interaction between these components. The genes we investigated are brain-derived neurotropic factor and cell transference 2. Brain-derived neurotropic factor is one of the most characterized neurotrophin for its role in brain development and in synaptic plasticity. Its expression is reduced by stress, and BDNF levels are reduced in the hippocampal and prefrontal cortex of postmortem measurement in suicide victims, as in serum BDNF levels are reduced in depressed patients relative to controls, but it can be normalized, as I told you before, by the antidepressant treatment. The most analyzed BDNF polymorphism is the VAL66 method. In here you can see that the literature say that the MET allele, which is the minor allele, is associated to decreased level of BDNF. And the stressful life events can increase the vulnerability to the depressive phenotypes in these carriers. Moreover, the carriers of the MET allele had reduced BDNF levels if exposed to childhood abuse, but higher than the VAL carriers when not exposed. So we can say that the MET allele, due to more sensitivity on the stressful life events. To our knowledge, no comprehensive studies were found on the SNPs we analyzed in relation to the antidepressant response, except for this SNP 11030101, but no significant association was found with clinical improvement. The other gene we analyzed is cell transference 2, which mediates polycyclic acid biosynthesis, which is involved in brain development, postnatal growth, and neuronal survival. Its expression is massive during brain development, but in the adult brain, this expression is reduced and retained in regions such as hippocampus, where it has been related to neurogenesis and synapses remodeling. In our sample, patients included were aged more than 18 years old. They met the DSM-4 criteria for mood disorder or anxiety disorder. They had an Hamilton rating scale score greater or equal to 8, and they were all treated with antidepressants. They were followed up for 4 weeks after antidepressant administration. Patients aged more than 85 with a concomitant diagnosis of mental retardation or neurologic disease or other medical illness that impaired the psychiatric evaluation were excluded. We enrolled 114 patients. The sample is composed by 65 females. The mean age is almost 50 years old. 68% of them were affected by major depressive disorder, 30% by anxiety disorder, and 2% by bipolar disorder, but with an Hamilton rating scale at the baseline greater or equal to 8. 40% was using benzodiazepine 2, 6% antipsychotic, and 7% mood stabilizer. This baseline Hamilton rating scale for depression score at the baseline was 13.5 with a standard deviation of 5.9. And these are the mean score of the stressful life events that we obtained. The diagnosis was according to the DSM-4 criteria and on the basis of an international neuropsychiatric interview. Sociodemographic and clinical variables were collected by a clinical interview. The patients were evaluated at the baseline and weekly thereafter for 4 weeks using the Hamilton Rating Scale for Depression. They failed also a modifying Brown and Harris stressful life events interview that includes childhood, onset, and post-month stressful life events. As a statistical analysis, we use the allelic analysis, and we categorize the stressful events by timing of the occurring event and by presence and absence of trauma. The analysis of covariant for repeated measure with the Hamilton score at the baseline was used as a covariant. The T-student test was used for evaluating the interaction between the genetic factors and the percent of the response. General linear models was used to evaluate the interaction between the alleles and the stressful life events on the HAM-D score, using always the baseline HAM-D score as a covariant or using the percentage of response. We use ApoView for test the Heidi-Wamberg equilibrium and the linkage disequilibrium. So, type analysis were performed by R software, and the estimation of the most probable haplotypes for each subject for interaction analysis was made by R software too. So these are the SNPs we analyzed. As you can see, some of them were excluded as not in Heidi-Wamberg equilibrium. Here you can see also the frequency of the minor allele in our sample next to the one in the literature. So the genotypes and the sociodemographic and the clinical variables were not associated except for the number of childhood stressful life events that was higher in subject positive for access to disorders. So we can say that an history of childhood stressful life events can increase the risk of the development of access to disorders in patients. And this could be perhaps a new point to investigate. Moreover, the age of onset was correlated negatively with the number of onset stressful life events. Then there was no association between the genotypes and the stressful life events. These are the interaction between the stressors and the 4-week outcome. As you can see, it isn't statistically significant, although you can see that the presence of stressor can due to a more severe clinical picture, and it's the same for the other stressors. These are the stressor at the illness onset, and these are for the current episode. So we evaluated the genetic effect on the 4 weeks outcome. We found only one result on this SNP for 777989, but we suppose it might be a bias effect, because as you can see, it's not relevant from a clinical point of view, because the two alleles differ just for 1 point at the HAM-D score. So we evaluate the interactive effects of the SNPs of BDNF and side transference of with the stressful life events. We had some results only at stressful life events occurring at the onset of the illness. As you can see, the carrier of the E allele of the SNP show a worse response, or better, a slower response at 4 weeks compared to the same allele carrier in presence of stressors. Moreover, the carriers of the T allele are not influenced by the presence or the absence of stressors. We had a really similar result in the SNP, always belong to the brain-derived neurotropic factor. As you can see in here, the G allele carriers has a slower response compared to the E allele carriers in absence of stressors, but they have a similar trend when they are in presence of stressors. So we used AppleView for test the ID-member equilibrium and the linkage disequilibrium. We had to exclude this SNP because it wasn't in ID-member equilibrium in our sample, as we had to exclude this haplotype GAG because its frequency was less than 5% in our sample. With the haplotypes analysis, we had a similar result to the allele one, because as you can see, the carriers of the AAG and the GAA haplotype show a slower response in absence of stressors. In here we focus on the two SNP, and the results are stronger than before, as you can see. Good. I forgot to tell you that in here the analysis of covariance for repeated measure wasn't statistically significant. Instead, in here it is. And so, perhaps the two SNP are in charge of this result. So, in summary, we found that the E allele carriers of the SNP 11030101 and the G allele carriers of the other SNP had a slower response to antidepressants if not exposed to stressors at the onset of the illness, whilst they have a comparable trend to the carriers of the opposite variant if exposed. The same result stays also in the haplotypes analysis. So the AG or the AA haplotypes have a slower response to antidepressant if non-exposed, whilst they are comparable to the other genotypes if they're exposed. And these are the results of the analysis of self-transference to SNPs. In here we had results only for the stressful life events at the onset again. So you can see that the carrier of the E allele in absence of stressors had a slower response compared to the same carriers allele with the presence of the stressors. A similar result was in this another SNP. As you can see, it's very similar, the trends. So we use AppleView to make a linkage disequilibrium map. The block 1 contains the SNP who gave us results before. But when we try the haplotype analysis, we can find a statistically significant result. Anyway, statistically significant in the analysis of covariance for repeated measure, but as you can see in this, with the percentage of response, it isn't significant. You can anyway see this trend of the AC haplotype, which is slower in absence of the stressors. So, in summary, the carriers of the E allele and of the 1185392 SNP and the carrier of the C allele have a slower response to antidepressant if not exposed to stressful life events at the onset of the illness, whilst they are comparable to the other carriers of the opposite variant if exposed. Anyway, we didn't find a significant difference in the AD and the antidepressant response between the haplotypes of these two SNPs. So, in summary, we can say that variants within BDNF type transference decrease the early response to antidepressants in individuals non-exposed to onset stressful life events only. According to our data, variants in BDNF and cell transference may so influence differentially the early response to antidepressant treatment depending on the exposure to stressor at the illness onset. No effect was observed on the most investigated VAS66-MET polymorphism. However, we found the results on two SNPs in strong linkage disequilibrium with this polymorphism. If, as I showed you, BDNF is very important for the antidepressant response, as all the animal studies shows, a recent study reported discordant findings, and the relationship between stress, BDNF levels, and the response to the antidepressant treatment is still debated. Studies on animals recently have shown that an overexpression, transgenic overexpression of BDNF may have an unexpected facilitatory effect on anxiety-like behavior and memory and learning impairment. A recent study in humans also found a better response to the antidepressants in the heterozygosis BAL66-MET polymorphism subject, and so perhaps also if BDNF may exert an antidepressant effect, too much BDNF might have a detrimental effect on mood. To our knowledge, no study investigated BDNF genetic variants on early response to antidepressants, taking into account the onset stressful life events so far, and we didn't find any evidence for cell transference as well. So, the first hypothesis for explaining our results is that pro-survival factors may be detrimental for early response to antidepressants in a depressive condition less dependent from environmental factors. In other words, the beneficial effect of pro-survival factors may be relevant only in stress-related disorders. And a second hypothesis is that the pro-survival factors may differentially interact with other systems involved in stress response. So, in some patients with some variants of these genes, the stress can help a faster response to the antidepressant treatment. The major limitation of our study is the small sample size. So, we can't excuse stratification bias leading to false positive results. We had also a low frequency of the minor allele in many cases. We had to perform the allelic analysis instead of genotype analysis. So we couldn't see the heterozygous effect. As you can see from the slide, some SNPs weren't tested because they weren't in high equilibrium in our sample. Another potential limitation is that we decided to focus on the early response. So we analyze a trend response and not the outcome. Moreover, the life events are retrospectively self-rated, and there wasn't an evaluation of their perceived impact on well-being, and there might have been also some recall bias. In conclusion, our data doesn't support the expected finding that the pro-survival factors favor the early response to antidepressants. At the opposite, we found genetic variants slowing down the response to antidepressants in subjects non-exposed to stressors at the earliest onset. So, we may hypothesize that in this subject there might have been an excess of pro-survival factors that interfere with the antidepressant's mechanism of action or other genetic factors when the illness is not stress-related. For the most sample size of our sample, as I told you, further replication or larger international databases are required. Thank you for the attention.